The overall goal of this experiment is to identify Zika virus-specific peptides for use in a diagnostic assay to detect Zika virus antibodies. This is critical as the virus disappears from the blood in approximately 10 days, while antibodies persist for years.
This method allows us to identify antigens and epitopes utilizing a microarray of the entire Zika proteome to identify peptides that capture either IgM or IgG antibodies in a sample of serum from an infected individual. The main advantage in detecting both early antibodies, IgM, and later antibodies, IgG, is to provide the clinician a timeline of the infection, and this is particularly important in pregnant women.
While this protocol provides insight into the course of Zika virus infection, it can be applied to other pathogens, using a microarray specific for that pathogen if its protein sequence is known.
Handle the glass slide by the edges using powder-free gloves. The glass slide dimensions are 75.4 mm by 25.0 mm and 1 mm thick. Place the slide in the incubation tray with the microarray surface facing up. Fill the empty slide holders with blank slides to prevent the microarray slide from breaking. Place the glossy side of the seal facing downwards onto the microarray printed surface. To ensure a good position, overlap the screw holes of the seal and the base plate.